Hi guys, Mr. Holding. This video is just covering nucleotides and base pairing. So when we've been talking about DNA, one of the questions comes is, well, what is DNA made of? And the answer to that is it's made up of what's called nucleotides. And nucleotides are made up of three things. Okay, first of all, you've got a phosphate group, and then a pentose sugar, and then this thing called a nitrogen base. Okay, and the nitrogen bases are those AT, C and G that you would have heard of before, and basically they connect with each other and they create that double helix effect. So I'm going to hand over to someone else who's going to explain this a little bit better.
But what are they made up of? Those are nucleotides, and so these are the building blocks of DNA and the building blocks of RNA. So let's look specifically at one. So this is one nucleotide right here. A nucleotide is made up of three parts. We've got a phosphate group, that's going to be pictured right here. It's in phosphorus in the middle and then oxygen around the outside. Phosphate groups are really famous in biology. So they're the phosphates that are found in phospholipids that make the cell membranes of all life. And it's the same phosphate that we're going to find in ATP, adenosine triphosphate. It's the energy source. And in fact, the adenosine triphosphate is exactly the same adenosine triphosphate that we add to make DNA. We'll get to that in just a second. What else do we have? Well, we have a pentose sugar. Pentose sugar means we have a 5-carbon sugar. In DNA, that's going to be a deoxyribose sugar, and then in RNA, it's going to be a ribose sugar. And then the most interesting part of a nucleotide is going to be the nitrogenous base. And it's called a nitrogenous base because it has nitrogen. And so most things in life are made up of carbon, but there's going to be a lot of nitrogen here in the base of this nucleotide. And this is going to be different in each nucleotide. And so let's take a look at the nucleotides found in DNA. And so basically you have adenine, cytosine, guanine, and thymine. And so we have 4 different bases, and therefore we have 4 different nucleotides. And you can just see, looking at them, the size is going to be a little different on all 4 of these.
Okay, so as you can see by my colleague Mr. Anderson, our nucleotide is made up of 3 things: a phosphate, a pentose sugar, and a nitrogen base. Now we're going to have a look now at how these nitrogen bases contribute to the bonding, and they end up with that unique pattern replicating how you see that codes for certain things. So we're going to have a look at that now.
So let's talk about bonding. How do you connect them together? Well, when we talked about carbohydrates, there's really only one way to connect carbohydrates. Or when we talk about amino acids, there's really only one way to connect them. But especially when you get to DNA, you can connect nucleotides in 2 ways. So let's start with way 1. Way 1, we can put this one right underneath it, so we got an adenine and a guanine. And then through a dehydration reaction, we could lose a water right here, and we could form a covalent bond between 2 nucleotides. And so if we were to add another one, we would add another nucleotide here, we lose a water, and we're going to make another covalent bond. And so we can attach them together like that. And so that's what RNA is. RNA is a number of nucleotides simply in a row, and they're connected with covalent bonds between each one. There's another way, however, when we get DNA that we can bond them. And so let's say we have these 2 nucleotides, adenine and guanine. How could I attach this thymine right here? Well, basically I can turn it upside down, and it's going to form hydrogen bonds here between the adenine and thymine. And you've probably heard this before, that adenine will always bond to thymine, and guanine will always bond to cytosine. And that's why there's going to be interactions between the oxygen, nitrogen, and the hydrogen, and make these hydrogen bonds that are going to connect the 2. And so when you're looking at DNA, let's kind of switch to this next slide. When you're looking at DNA, that's what's being connected right here in the middle. So that's going to be those hydrogen bonds between the nitrogenous bases on either side.
Okay, so now we know that we've got our DNA, or our RNA for that matter, is made up of a phosphate, a pentose sugar, and a nitrogen base. Okay, we've got this backbone, which is the sides of the ladder, on this side and this side, that's made up of the phosphate and the pentose sugar. Just incidentally, the pentose sugar can either be deoxyribose, which is a sugar, or ribose nucleic acid, which is a sugar. So that determines whether it's DNA or RNA. But in DNA, this would be deoxyribose. That's our backbone, and then the rungs of our ladder is formed by these nitrogen-based pairs. Okay, and C and G, so that's obviously upside down. Okay, you can see it this way: A, G, T, and C, whichever way you want to put it. Okay. we have it going that way. Now on this side it heads in that direction, and this is the opposite. This way it heads in that direction. Okay, but ultimately we end up in a situation where we have these nucleotides pairing off, and that's ends up how we end up with the backbone and then the individual rungs. Okay, and that in itself then, as it gets twisted, and that creates that compact double helix situation. Okay, so one final thing there to remember is the pairing rule. Okay, and easy way to remember is C and G look like each other, and so they always pair. That means the leftover is A and T. So all you got to do is remember one pair. Okay, there's only 4 of them, C, G, A, and T. There's the only 4 bases, and they only pair with one other. So C only pairs with G, and A only pairs with T. So if you just remember C and G are like each other, so therefore they pair, you can work it out for yourself. So hopefully that gives you an explanation of the nucleotides and the base pairing rule. Okay.